Thanks everyone for coming. Let's get started. How's everyone going with their projects? Good? Yeah? Anyone want to throw out any questions or any problems, issues? Everyone's groups are all buddies and everyone's working well together. No infighting. The factions have formed and you're fighting for power. No? Okay. Good. So the critical reviews will be marked by the end of this week, and you'll get your marks hopefully before the start of next week, and then you'll have a chance to go through with your tutor on your tute next week. So sorry that's taken a bit longer. Just a few teething problems for the first one. So I guess I'll move on to the lecture. So last week we spoke about imaging in general, a bit of image formation, lenses, and then also some sensors. So how do you detect the image, particularly digital sensors, CMOS and CCD. So today we'll talk about, one of the biggest fields for biological imaging, microscopy. This is a microscope image, 150 times magnification. Thanks guys. And this was taken, does anyone know the type of microscope this was taken with? Nope. It's called a dark field microscope. Most of the really cool images you'll see are either fluorescence or dark field microscopy. Thanks. So we'll learn about this today and a bit more fundamentals about how microscopes work and talk a lot about resolution and what resolution means. So learn about resolution, sensor resolution, optical resolution, magnification. So the microscopes are basically just magnifying apparatus. Numerical aperture and what that means for resolution. What else? How we measure resolution. I'll do a bit of a demo, and then we'll talk about, after halfway, we'll talk about some types of microscopes, some practical stuff, how they work, what they're good for, and some examples. So resolution. Does anyone know what resolution is? No? Something you do on New Year's Day? No? Sorry? Correct. Yes. In, more general layman's terms, it's basically the smallest thing you can see. So what can we resolve? What can we actually see with our imaging system? Sorry, I should have put you on here. So, what can we see? Why is it important? Well, we need resolution. We need to be able to see small things in biology because a lot of biology stuff happens on small scales. So this is an image of blood. See red blood cells, and that's a white blood cell. And sort of that cellular level is where a lot of biology happens. And so these are usually sort of 1 to 100 micron in size cells, biological cells. So red blood cells are about 8 micron in diameter, their largest side. So we want to be able to see these things and image them so we can see the processes going on in the body, all sorts of applications. So we need to be able to resolve small things. We need to be able to see things that are very small. So optical microscopy allows us to directly image cells and other small objects. Basically, we use high magnification to push the limits of spatial resolution. So we want to see this. We want to really push those limits. So we need to understand resolution, so we know how our imaging system's working. What influences the resolution? How can we improve it, and how can we measure it so we know what we're actually seeing? There's a few examples there. Anyone want to guess what the bottom left is? Nope. Anyone? Sorry?
Is it a tapeworm?
A what, sorry?
Tapeworm.
Is it a tapeworm? The bottom right is a tapeworm, yes. Bottom right's a tapeworm. Really disgusting. Bottom left is a daddy longlegs eye. Imagine how tiny that is. The top right is it's not Pac-Man, it's an algae, green algae, an algae cell reproducing, so releasing its daughter cells. And the one in the middle that looks like a Christmas tree is actually stem cells growing, I think it's bone, fatty tissue. So some of these, there's these beautiful images, you can get them on the internet. There's a sort of competitions, the Welcome Image Contest has a lot of these kind of things, if you want to look that up. And also the Nikon microscopy, that's where these come from. Every year they kind of send us your best images and they put them up. A lot of them are false coloured, so they don't actually come off in colour off the microscopes, but then you can false colour them to make nice images. So let's talk about microscopy a little bit. This is probably one of the best known early examples of microscopy. It was this book by Robert Hooke, so 1665, that's a very long time ago. That's the microscope that he used. You can imagine 1665, have you ever seen a flea in real life? You can't see much, just look like a little speck. But you look down a microscope, you see this kind of thing happening. He did these beautiful illustrations. There were not really photography around very much. And imagine the world just opens up a whole new world. You know, this would look like a monster, really, but it's a flea. No one would have seen that before. That's pretty amazing. And so that's sort of how long microscopy's been around for. Obviously, it's changed quite a bit. And mainly it was just looking down the microscope, maybe you draw what you see. Now we want to actually use imaging, particularly digital imaging of that. So what is resolution? It's the smallest feature that can be seen in the image. So more technically, it's the minimum distance between two distinguishable objects. Typically, we use units of line pairs per millimetre. If you think about what that is, how many lines can you fit into 1 millimetre that you can still see? So more line pairs per millimetre means smaller lines. And our application defines what we require out of our imaging system in terms of resolution. So here's two images of blood. On the left, we can't resolve the blood cells. You can see them, they're red, and this is where the kind of the subtlety comes in, and we can see them, but we can't distinguish them. We can't see two, we can't tell that there's two in there, we can just see red. But if we wanted to do a blood splatter analysis, this resolution is fine. Resolution of a few millimetres maybe. If we want to see the red blood cells and see what's in them, we get what's on the right. Use a microscope, we can get resolution of sort of microns. And you can see the difference there of what we can actually make out. So if we don't need to see the cells, we don't need to have that resolution. So it's really important to know what you're wanting to look at to make sure you have enough resolution to see what you want to see. Okay. So what influences resolution? Essentially, most of the parts of the imaging system will influence resolution. There's two sort of main factors with your resolution of your imaging system. The first one there is sensor resolution. So this is talking about a digital sensor. Essentially, if your pixels aren't small enough, you won't be able to see things. So the limit is, I'll go into this in more detail, but basically your effective pixel size, what each pixel represents in real space, the size of that sets a limit to your resolution. If it's too small for your pixels, you can't see it. And you can see there a bit of a, you'll see why I put that up there later, but that hole's getting pretty close to being not resolvable. And actually the smallest scale that you can resolve, where two objects are distinguished, would be 2 times the effective pixel size. We'll talk about that a bit more later. The other kind of factor is optical resolution. So if you have a point source of light, an infinitely small point, when you pass that through your imaging system, lenses, magnification, you don't get a tiny little point on your image. What you get is a point spread function. And so you can imagine that kind of sets a limit on the resolution. If you have 2 points that are very close together and they come out as an airy disk, which is what you see there, they come out as a blurry sort of circle, then you won't be able to distinguish them if they're smaller than that circle. And that point spread function is largely determined by the numerical aperture. And again, I'll go into this in more detail. So they're the two we'll talk about today. And they're the two sort of main influences on your resolution. Okay. So let's talk about sensor resolution. Sensor resolution, essentially it's based on this, what's called the Nyquist sampling rate. So there's Harry Nyquist. He was around in the sort of turn of the last century there. And he was a mathematician, and he basically came up with the theory that for any signal, to avoid what's called aliasing, you need a minimum sampling rate of 2 times the highest frequency. So if you think about a signal that's a sine wave, sine waves have a frequency. If you want to actually sample that with discrete measurements, then you need those discrete measurements to be at a sampling rate at a frequency of 2 times the highest frequency in the signal that you want. And there's an illustration of this here. So essentially, we have those black dots are our samples. So imagine you're sampling a signal, and you've got all those samples of your signal, those black dots. Now I can put 2 different sine waves in there that coexist with those dots, co-locate. So essentially they're both valid signals. If you have that sampling rate of those black dots, you can't resolve that red signal. Basically what you're resolving is the lower harmonic of that. So there's 2 different sinusoids that fit that same set of samples. So the sampling rate isn't sufficient to resolve that red sinusoid. So essentially, your pixels aren't small enough to resolve that signal. You can't see it. And so what does this mean for imaging? Well, basically, same as I said before, features in the image, think of them as signals, as spatial signals. So if you think of a Fourier transform, you can decompose any signal into sinusoidal functions. They'll have frequencies. High frequencies are the small things. Low frequencies are the large things. And what you're doing is sampling that whole space in discrete locations that are evenly spaced. So you need the sampling rate, the sort of the spacing of those discrete samples to be high enough, to be small enough, to be able to resolve that highest frequency. And essentially you need two pixels. So this is a synthetic image of a disk. You can see it's pixelated, looking really small. And so it's the disk is 36 pixels wide and the hole is 12 pixels. And we can resolve it. It looks like a washer, say. Now, if I then make this smaller, now the disk is 9 pixels and the hole is 3 pixels. We can still see the hole. We've resolved it. There's 2 pixels there. We can distinguish that hole. It's greater than 2 pixels. But if I then make it even smaller, now I've got a 3-pixel disk. I can see that, but I can't see the hole anymore. It's smaller than a pixel. Well, it's smaller than 2 pixels, so I can't see it anymore. It's below my sensor resolution. And that sets a lower limit on what you can see in your image with a digital image. Okay, does that make sense? So the take home basically is that you need small pixels, and you need 2 pixels across any object to resolve it. So the effective pixel size determines the sensor resolution. The effective pixel size is the size that your pixel represents in real space. And the effective pixel size is related to the native pixel size. Remember, that's the actual physical size of the pixel on the CCD chip or the CMOS chip. And that is related to the native pixel size by magnification. So we've got effective pixel size equals the native pixel size divided by the magnification. If I have a magnification of 1, then every pixel in my image represents the same size of pixel, the same in real space. If I have magnification of 2, then my effective pixel size is smaller than what it represents in real space, because I magnified it. So what can I do to improve my sensor resolution? 2 things. I can change the pixel size, native pixel size, sorry. So I can get a CCD chip with smaller pixels, or I can increase my magnification. So magnification will improve your resolution, kind of intuitive. But there's limits to what you can do. So you can't increase it infinitely, or it's very difficult to. There's the equation up there for magnification in terms of the focal length of your lens. So basically what you need is a very high power lens with a short focal length. It means you'll have a very large image distance from your lens, and it becomes impractical. You need a very large lens. So you can only go a certain distance with that. But, we can do it. We do microscopes, that kind of thing. So if you want to improve your sensor resolution, you can increase your magnification. Okay. So that's one side of the limit of resolution, is your sensor. So what about the other side, optical resolution? And this relates to your lenses that you're using. There's this thing called the diffraction limit. So resolution depends on how well a point of light, as mentioned before, is transmitted by the optical system onto the image plane. And you have what's called the diffraction limit, whereby you cannot no longer resolve that point. So it gets small enough that you can't resolve it anymore because of diffraction. And I'll explain that in a minute. So you can increase your sensor resolution, but it doesn't mean you can resolve smaller things necessarily. So you can increase magnification, but all you're doing is like oversampling a blurry image. So you don't actually see anything new, you're just seeing a bigger blurry image, essentially. And the diffraction limit, or the point spread function, is determined by the numerical aperture. And this is a dimensionless number that characterizes the range of angles over which the system can accept or emit light. So if you look at that image there, you can see that angle. Basically, it's that angle. So if you have a very powerful lens, a very large lens, that's usually a large numerical aperture. If D is very large, you have a large numerical aperture. Okay. So what's this point spread function thing? So basically it describes the response of the imaging system to a point source or a point of light, or a very small object. And what you essentially get if you have something that's very small, that's beyond the diffraction limit, is an airy pattern. It's a bit hard to see there, but there's actually rings coming off that. And so that is what a very, very small object will look like on your image. So why does this occur? I'll play these. So it's due to diffraction. You all know what diffraction is. Basically, if you have a very small hole and you shine a sort of a laser light through it, it will diffract and you'll get this kind of cone coming out. The size of that hole, the size of that diffraction pattern will depend on the size of the hole, size of the aperture. A larger aperture will have a tighter pattern, and that's due to interference. This is real. It's probably a bit too much detail, but this is an example there of that diffraction happening there. That's a plane wave coming in, a straight wave, goes through that aperture, and we can see this pattern here, this diffraction. So basically, the influence on resolution is that you now have these disks rather than a small point of light that you're trying to image. So if the point spread function is large, you have large airy disks, and objects must be further apart. So on the bottom left there, we have these airy disks here. So this is an image, say, think about it, an image of a star. That's what you'll get. If you have two very close together, they'll start to overlap because it's no longer a point, it's sort of a larger. And again, numerical aperture. So if we increase our numerical aperture, increase the size of the lens, we get a smaller airy disk. So we can more accurately transfer that point source of light into an image. And with that comes increased resolution. Okay. So that's basically it for resolution. There's sensor resolution and there's optical resolution. So how can we improve, we've spoken about optical resolution, how can we improve optical resolution? Well, the area disk diameter is given by this equation here. D equals lambda over 2 times n sine theta, where D is the diameter, lambda is the wavelength of the light, and n sine theta is Na. Remember that first equation. So n is the refractive index of the medium. So most cases that's air. And then there's the light gathering angle, which is a function of your lens power and diameter. So in practice, For optical imaging, for a sort of a normal lens, you can get basically lambda on 2. So if you think about light, somewhere sort of green light is 500, somewhere like that, 500 nanometers. So we can resolve down to 250 nanometers. That's the size. So how can we prove it? We've got this equation. What do you think? What can we do? These are the parameters that are determining our optical resolution. Wavelength, refractive index, and light gathering angle, or NA. So what should we do? Increase NA. Yes, definitely. If you increase NA, you get a nice big lens, then we will get better resolution. And so if you have, the really powerful microscope lenses will have a very high NA, very expensive, because it's hard to make a lens that big that's quality enough. But yes, NA, and that's your first port of call. What else? NA is, actually, let's go here. Okay, so increase sine theta, increase NA, basically. Sine theta is limited though, 1, right? You can't get bigger than 1. So even with a very big and expensive lens, it's difficult there's a limit to how much you can increase your NA. Essentially, it becomes too expensive, and also there's actually a fundamental limit. So that's kind of, you can push that only so far. So what else can we do? Increase N, increase the refractive index. And this is what we do. So really high-powered microscopes, we'll use an oil immersion lens. So essentially, we'll replace the air gap, which has a certain refractive index, with oil. And that oil will have a higher refractive index, which means that our D, our area disk, will be reduced. And there's a picture of that on the top right there. You can just see at the bottom there's sort of like a pool there. That's our microscope slide. That's our lens coming down. And you basically put a drop of oil there, and then you place that there. So now you're imaging through oil rather than through water, and that gives you a higher possibility of resolution. And the other option is to change the wavelength of light. So visible light obviously is a certain range of wavelengths, but we can use different parts of the electromagnetic spectrum. So X-rays have a wavelength which is 1,000 times shorter, and X-rays can image at much higher resolution than visible light for this reason. And electrons have an effective wavelength that's 100,000 times shorter. So you've seen that's an electron micrograph, so electron microscopy, and that's 3,000 times magnification. So this is where you see these images like this, and they're sort of, super high resolution because we're using electrons. And the wavelength is so much shorter, so you can see things that are so much bigger. Sorry, at high magnification. All right. So that's basically the fundamentals of resolution. Now, how do we measure it? Anyone think of how we might measure our resolution? Sorry? Yep. So what if we just take a picture of a really small thing and see if we can see it? And that's what you do. So here's an example of a resolution target, and this is the standard, it's a 1951 standard US Air Force resolution target on the left. Essentially what you do is take an image of something like this. So this has a lot of line pairs, and they are decreasing in size, getting closer together. And so you can see ones you can resolve them, and ones you can't, and that will give you your limit. They use these to test also resolution of satellites and sort of spy planes. So you can see on the right, that's a giant resolution target in the middle of nowhere, America, and it's got a resolution target on there, so they can test the resolution of their camera systems, their spy cameras and stuff like that. So that's pretty fun. All right, so what do? We use the line pair phantom. So it allows us to see the limit of our resolution. And the smallest line pair that we can distinguish gives our resolution. That's a kind of qualitative thing, I guess, because we can. It's not binary. You can't resolve it or not. You can resolve some things better than others. But it's a good first go. But what's even better is to do something more quantitative, which is what's called the modulation transfer function. So what we do is plot the modulation of those line pairs, where modulation is essentially how accurately the modulated signal, which is the line pairs, is transferred to the image. There's some equations there, but I'll just show you over here. So if we have, it's related to the point spread function. So if we have a point spread function there on the left, so remember this is what a point of a very small object will come out as on our image. If we have an edge, which you can think of as the edge of your line pair, so we have very dark and then very light. And at every point along there, you have essentially transferred as a point spread function. What you get is what's called the edge spread function, which is this here. So this is what a sharp edge will look like in your image. And you can see how that's related to the point spread function, which is again related to your optical resolution. So that line is not resolved. Well, it's resolved, but it's not, it's obviously not sharp. And if we put a whole lot of these together, we have here this. So these are your line pairs. Think of these as your line pairs. So we have very dark intensity and we have very light intensity. And if we image this, we don't get that perfect modulation. What we get is this. And the modulation transfer function relates to the difference between the light and the dark intensity here and the light and the dark intensity here of what you actually get compared to what you have tried to image. And as we get smaller and smaller in our line pairs, closer and closer together, that transfer is less efficient, think of it like that. So basically we get less of that modulation transferring through to our image. We get smaller again, you can see this modulation here, which is what we get in our image is actually, less than half of what we've tried to image. And so if we plot this with frequency on our x-axis and modulation transfer, the percentage of modulation which actually is transferred, we can get an idea about how resolution is affecting our image. So essentially what you do is you take an image of a resolution target and you plot the modulation transfer of all the line pairs at the various scales. So we get a plot like this. So at very low spatial frequency, which is very large line pairs, we get perfect transfer of the modulation, 1. And as we get smaller, as our frequency increases, our line pairs get closer and closer together, we get less and less. At some point, you're not really resolving them anymore. You might get some aliasing, but you're only getting a certain percentage, 10% of the modulation coming through. And so essentially what you do is pick an arbitrary threshold, say 0.2, and you might call this MTF 0.2, and then you pick what's my spatial frequency that I get that modulation through, and that might be your metric for what your resolution is. So the spatial frequency, lines per inch, lines per millimetre, is your resolution based on that metric. 20%, 10% is often used as well. Okay, so does that make sense? That's a bit. Anyone have questions on that? Okay, I've probably lost all of you. That's okay. All right. So what I want to do now is give a demonstration of how to measure, or how we might do it. So this is a live demo. I haven't tried this yet. I printed a resolution target. And I've got my little microscope here too. So let's try this. All right. All right, cool. And I think I did the lights too. All right. So what I have here is a line pair for Anton. You can see it's got line pairs coming down. It's in blocks, and each of these is a standard size in real terms, real size. And so we've got like the first block here, smaller and smaller, and then you have, so 1, 2, 3, 4, 5, 6, and then you have a second block here, 1, 2, 3, 4, 5, 6. And these are like logarithmically descending in terms of their size. And then we have, 1, 2, 3, 4, 5, 6, Probably you can't see that. 1, 2, 3, 4, 5, 6, getting smaller and smaller and smaller and smaller to basically to infinity. So let's see what we can do with this. Now you can see, obviously I'm holding it so it's not great. As we get smaller and smaller, probably that 1 there you see in the middle, and then 1, 2, 3, 4, you can't even see the numbers anymore, that's probably the limit. So I'm going to write that down. So this one is probably, I think it was this, was it? So this here is probably the block where my, not that one, sorry. This one. That's the block of the limit of my resolution for this system. You can just probably make out the number ones there. I don't know if you can see it up there. No, not very well. Okay. So basically that's what you do. What do you think is going to happen when I put my microscope on? We'll go right down to the middle. So let's have a look. So I can now increase my magnification. I'm increasing my NA, as we figured out before. So essentially I'm putting a high-powered lens on. I'm increasing my NA, which is going to give me A, better sensor resolution, and B, better optical resolution. Now let's give this a I need my little thingy-majiggy. All right. So I haven't actually tested this, so we'll see how we go. What's that? What's this? Sorry. Nope. Why is that? There we go. All right. This will be fun. Yeah, so that's what we could see before. You can see they're all really well resolved now. Now if I get even better. And we can see block, next block down, and actually we can see I've actually hit the limit of my printing. This is a university printer, it's a pretty good printer. Can't even print small enough. Okay. So maybe that block, block 3 there, it's maybe the limit, but it's hard to tell because of the printing. So remember, I've gone really close here. It was a lot harder to get focused. This is auto-focusing as well probably, so that's, but my depth of field is a lot smaller. And my resolution has increased by each of those is a factor of 10, so 100 times more. You can buy these sort of laser etched and that kind of thing, so you can get in smaller. Okay. Cool. So that's my demo. And that's what I want to talk about for resolution. So if you guys want to come and have a look at this, at how small that actually is, I don't know, but we'll have a break, we'll have 5 minutes, and then we can talk about some applications. Cool. Thanks. Or do you want to just keep going? Get out early? Yeah? All right. Keep going? All right. All right. It might be a short lecture today because my other demo isn't going to work. All right. Anyway, I'll talk a little bit more. So when you pick out a microscope, you might actually get this, and I encourage you to look these things up. You'll get an MTF function. That was my demo. So, all right. So I'll talk about a couple of different types of microscopes now, and they're kind of the most simple and the main ones. I'm not going to go into too much detail about the really sort of more complicated types of microscopes. I'll talk a little bit about what they're good for, what they're usually used for. And then, as I said, I'll give you a bit of bit of a case study of something that they're used for. So this is a bright field microscope. Hopefully you can see that. Great. So bright field microscope, this is probably what you used in high school, sometimes called a student microscope. You can buy a very simple, very, not a very good one, but for like $5 at Kmart. Don't do that, they don't work very well. Spend a little bit more. But essentially what you're doing is you've got transillumination. So it's the same as this, essentially. Or it's actually a little bit different. So you've got the light coming from one side, transillumination, that's here. You've got an illumination source, and you've got a sample, usually on a little slide, and you're imaging what is transmitted through the sample. So that's the attribute that you're looking at. You've got a sort of little slide here with a specimen holder there, and you have a lamp, some sort of light source. You have some lenses, condenser lenses. So this is to focus the illumination source. So you've got a bright, a bright source. You're taking all that light and focusing it down. So you've got a bright source or bright illumination on your sample. And then you've got an objective lens, which is what's actually imaging the sample. So this would be your objective, something like this. And then in this case, it's just got an eyepiece. But most microscopes now, you pull that off, put a camera on. So these lenses are essentially just magnifying everything and creating a sort of an image over here that you can look at. This is where most of the magnification takes place. Because it's a very high magnification, you've got a very short object to lens distance. This is true for all microscopes. Short object to lens distance, and then a very long lens to image distance. And so you need some of this stuff as well to help sort of get that within practical limits. You need bright illumination. You can get that through a lens, condenser, and you use some sort of pretty high-powered lamps as well. But essentially, you're shining a light through something, and you're trying to image it. So you need something that's pretty thin or semi-transparent. And if you remember when we spoke about magnification, NA, and depth of field, we've got a very high magnification, high NA, relatively high NA, so you've got a thin depth of field. So if you want high magnification, you need a thin sample. Otherwise, you're trying to image a section through all this other stuff, or this other tissue or whatever, which is going to be defocused. It's just going to make this blurry image. And you'll see sort of examples of that when I show you the case study. But essentially, you usually have a thin sample. If you have a very thin sample, you need that to get the light through, and so you don't have all this out of focus stuff in your image. But what that means is that it's going to be very transparent for a if it's very thin, it's going to be very transparent, and you might not get very good contrast because it's not absorbing a lot of light. So what you often do is stain your sample. And you probably did this in high school chemistry or in high school biology. Get a little thing, put a little bit of stain on there. So common applications is histology. Histology is basically you get a sample of tissue, maybe lung tissue. This is lung tissue. You fix it. So you put it in a chemical which turns it into plastic or hard, fixes it. Then you section it, you slice it really thin, essentially with just like a meat slicer. And then you stain it. And you can use stains which stain certain structures preferentially. So this has a stain which turns the nuclei of the cells blue. And the cytoplasm, which is all the other stuff in the cell, it's pink. It might be a bit hard to see up there, not too bad. And this is lung tissue. So this is where the gas exchange takes place. So these are very thin. These are sort of probably 5 micron, something like that. I should probably actually go to full screen, make it see it better. Yeah, so this is air. In a bright field image, the background is bright because you've got the bright light transillumination. And you can see these are the cells, nuclei, so they're all individual cells. And you've got this kind of honeycomb or foamy structure. Air comes in, you've got blood coming through here, and you get gas exchange across those thin boundaries. There's probably a little bit of blood there, maybe. Yeah, so that's a very, very common application. And what that allows you to do is see the macrocellular structures of things. So you're looking at how all the cells fit together. You can look at different types of cells and how they all form the tissue. Often also we get this sort of biological imaging. These are the simplest microscopes to use. bang your sample on, have a look. And so this is actually a mosquito head. So mosquitoes are pretty small. You can see all the structures there. They're small, so they're thin enough to actually see through, and you can get a bright field image. And you can see some anatomical structures there. Yeah, so that's the most probably common and simplest microscope is a bright field microscope. Okay, so what about this one? Dark field microscope. I spoke about dark field. That's the one with the thistle seed. So how does this work? Well, essentially, it images the light scattered by a specimen rather than the light that's transmitted through a specimen. So if you think about specks of dust in the air, and a ray of sunshine coming through, you can see that little bit of dust is actually scattering the light, scattering the photons coming from the window, say, and you see the scattered light. You don't actually see the light that's coming through. You're not looking straight at the sun. You're seeing the light that's scattered by the sample, and that's essentially what we're doing here. We're seeing just what's scattered. And this can improve contrast over bright field because the scattering is stronger. The differential scattering between different parts and different structures is stronger contrast than the absorption, especially if it's unstained. So the difference between the absorption of two sort of adjacent structures will be less than the difference between the light that they scatter. And so you can get improved contrast. It's really good for unstained biological samples, and that's typically what it's used for. So it means we don't have to stain things, we don't have to fix them. It means we can sort of look at living things as well. And often they're sort of aquatic samples. You're only seeing what's scattered, so that's pretty, I guess that's a small proportion of the light that you're illuminating. And so you need a very strong illumination. Much stronger than in bright field. And if you're, hammering this thing with light, then you can often damage the sample. So you need to be careful of that. So this is how it works. We have a light source. Think of this as a cone. It's a 3D cone. And essentially we put a patch stop here. So we're blocking out all the light that's going through the centre there. I've got just this light coming here. It comes through the condenser lens and is focused onto the sample. But there's this dark spot in the middle. So this light comes through the sample. Here it will scatter. Some of the light will scatter, some of it will pass through. And we have then the direct illumination block. So what that means is any of the light which is directly illuminating the sample is blocked out. So these are kind of, complementary, a patch stop and an illumination block. And so all you see is this yellow light, and that's the light that's scattered. It's going in a different direction than the direct illumination. So all you see is the scattered light. So you can see you're blocking out most of your light, and all you're seeing is this kind of scattering of light. And you get some, these nice images like this. So this is algae undergoing cell division 200 times. And this is a zooplankton there. So you can contrast this with the mosquito image that I showed you before. Background's dark because there's no direct illumination. All you're seeing is like the light scattered. And you can kind of see that here. You can imagine what it's like if you're looking from the side. The light's coming from another side. All you're seeing is what's scattered off there. And you can see a lot more of the internal structures. And you get much better contrast. This one's a bit out of focus, but you can see these kind of structures here, which is quite different to the other image. That's that website there too, so look that up. Okay, so perhaps the most powerful microscope technique is fluorescence microscopy, and I did want to do a demo of fluorescence. I'll do it next week because my UV torch has been misplaced, so I can do it next week. But essentially, fluorescence is what's used, most commonly now in biological imaging for various reasons. So what is fluorescence? It's the emission of light by a substance that has absorbed light or other electromagnetic radiation. So I have a substance, I have a sample, and I illuminate it with light. The atoms absorb that light, they absorb that photon. Remember, it's just energy. They absorb that energy, and then they emit another photon. And that photon has a shift in wavelength because that process loses a little bit of energy. So and I've got a photo of this later. I have a sample, I have an atom. Light comes in as a photon, as energy, I absorb it, and then I emit another photon which has a lower energy. Now, why do we want to use this? Well, essentially, we can control that emission. So we can tag different types of tissue to fluoresce at certain wavelengths. And what that means is we can differentiate types of tissue very easily, and we only image the emitted light, and so we know that that is what we've tagged, essentially. One of the really big kind of boons of fluorescence microscopy is that we can also make, certain genes, we can make proteins that fluoresce, and we can make them express at the same time as certain genes are expressing. And so we can essentially image gene expression. And that's hereditary, so that will pass down. That can pass down through generations, and that's pretty powerful. Cool. So how does it work? It works by the process called Stokes shift. So essentially we have blue light coming in, light of a certain wavelength, blue photon come in, gets absorbed. I missed something here. So, okay, sorry. This is an electron, and I didn't do absorption yet because I was saving that for next week. But essentially this is an electron in an electron shell outside your atom. Hopefully you remember this from physics. So that electron can absorb energy and jump to a higher energy state. So that's what it's going to do. This electron in the atom is going to absorb the energy from this photon and jump to a higher state. It gets excited, it goes to a higher energy shell. It's gained a little bit of energy. Now that's made that atom unstable though. This is going to relax a little bit. We're going to lose a little bit of energy to heat and torsion in the nucleus. And so we're going to lose a tiny little bit of energy. It gets lost in certain ways, and then now I'm unstable. Now I just want to go back to my ground state. So this electron is going to fall back to its ground state, and it's going to emit light. It's going to emit the energy that it gained as a photon, or the energy that it's losing here to go back to its ground state as a photon, and that's going to be green light because it's a lower energy. And that shift, that change in wavelength, is called the Stokes shift. And I've just been, sorry, I didn't get any sleep last night. So essentially, I'll go through that all again. Blue light, this is an electron. Feel free to call out if I'm doing something wrong. So we have an electron here, which is an electron in an atom. You can now see that. It's in its own ground state. It's got a certain energy. We're going to absorb some blue light. This is a blue photon coming in. It's got a certain amount of energy. This electron can then jump to a higher energy shell in its electron, in its atom. Now it's got this extra energy. Go back. It's got this extra energy. It's going to lose a little bit of energy just due to heat inefficiencies in the process. There's a bit of torsional energy going into the spin of the atom. So it's going to lose a little bit of energy, and then it's going to be unstable. It's going to fall back to its ground state, and it's going to emit a photon, and it's going to emit a photon of a lower energy. So it will emit green light. Okay. This process occurs because there's a specific jump in energy shell that this electron can take, which is equal to the energy of this blue light. So you need this exact wavelength to cause this fluorescence to occur, and it will always occur with this exact wavelength as the emitted light. And I'll do that demo next week. So this is really important because, thanks guys, because what it gives us is the ability to filter out those very discrete wavelengths of light. So if we have a white light source coming in, this is how our fluorescence microscope will work. We've got a light source, white light. We have what's called an excitation filter. This will filter out everything except for the blue light. So now we can have that incident on this dichroic mirror. This mirror will reflect certain wavelengths and pass through other wavelengths. So this will reflect blue light. We'll reflect that light through the objective lens down to the specimen. So now I'm illuminating the specimen with blue light. So that's my excitation or my illumination frequency. This will then fluoresce, and it will fluoresce green light. And this is really handy because that means I can use my dichroic mirror to block out any of the light scattered, any of this blue light that's coming in, the illumination, and just look at what's fluoresced. And so in that way, all I'm looking at is the fluorescing structures within my sample. And remember, I can tag those. So I can tag the sample to only have certain structures or certain proteins to fluoresce. So that's one method. Another method is using a laser. Essentially the same thing. However, instead of having a white light source, I just have a laser beam, which is a certain wavelength of light, 532, say. I have my dichroic mirror, I have my sample. This particular fluorescence process or protein will fluoresce green light, will take green light in, so it will absorb green light and fluoresce red light or orange light. And so that's just another method to do it. Okay. All right, so that's the basics of the fluorescence microscope. And why is this good? Well, I can use different labeling methods to label certain specific structures in my sample. And this is where the real power comes in. So there's this thing called DAPI blue, and that binds to DNA. And so what that will do is allow me to label just the nucleus of cells. I can also stain, say, just the actin fibers. This is the muscle fibers in the cells. So they're very important fibers within a cell as to how cells divide, all that kind of thing. And I can label just those. And they'll fluoresce at certain wavelengths, so I can just image those structures. And one of the big things is what's called GFP, green fluorescent protein. And this comes from fluorescent jellyfish. And what this allows me to do is to I can tag my sample to fluoresce green when a certain gene is expressed. So say I want to check a gene and see if this gene is to do with, say, heart disease. I can then tag my sample, my mouse, say, and then I can image it and see if that green fluorescent protein, where it is, and that will show me with a tissue where that gene is actually expressing. And then I can look at that and look at some kind of outcome of that gene, of how that gene is working in that tissue. Okay. And this is such a big deal that they won the Nobel Prize in 2008 for the development of the protein. And this is used all over. It's pretty important. So, some limitations of fluorescence microscopy. You get what's called photo bleaching. That stokes that fluorescent process. It actually can damage the molecules, and it limits the fluorescence exposure time. So if you hit your sample with the excitation frequency of light, it will fluoresce, and then that has a time decay. So the brightness of that fluorescence will decay over time. And that's a limit because, you can't just have infinitely long exposure times because your fluorescence would just disappear. The fluorescence process results in limited light coming out. It doesn't glow very brightly, essentially. So you need a large NA, you need a large lens to gather in all that light. You need sensitive cameras. Usually these are CCD cameras that you use for fluorescence imaging. And also, this is an advantage and disadvantage. Only the fluorescently labeled structures are imaged. So you might just get a blob of glowing something and you don't know where it is. You don't know what it's doing. You need to have some kind of reference of your sample to see what actually that structure is and where it sits inside your sample. And so sometimes you'll combine a microscope and you'll have a bright field image in there as well. And that allows you to sort of place that fluorescent structure within your sample. So just a couple of applications. This is a widely kind of shown image. You'll see this a bit. And this is a skin cell, essentially, endothelial cell. This has 3 different structures labelled. It's got the nucleus of the cell labelled with the blue DAPI. It's got the microtubules with a green, that's the green fluorescence there, and it's got the actin labelled in red. And imagine if that was a grey image, which I don't have here, but you wouldn't be able to make out those structures. It would just be mush. This is actually built up as a 3-channel image, and typically these are done. Each fluorescence is done sequentially. So you'll take the blue essentially. You'll hit it with a certain wavelength and fluoresce the blue light. You'll image that with a monochrome camera. Then you'll swap your filter, your emission filter, sorry, your excitation filter and your emission filter, and then you'll get the green and the red, and then you can build a fake, a falsely coloured image of those three channels built up together. And I've already shown you this K-fish. So this is neuron cells, and they're expressing that GFP. So we can look at congenital diseases because the green fluorescent protein is tagged to the gene, and that's hereditary. So you can look at how genes are passed down and how they affect the growth and development of an animal. Okay. All right. So let's have a look at a bit of a case study. Zebrafish. Anyone have an aquarium? No? Okay. Zebrafish you can buy in a fish shop. And the thing about these is they're basically clear up until about this age, about 21 days. And that's really nice for optical imaging because we can image them very easily. They're pretty, they have a sort of pretty short reproduction cycle, and they're pretty easy to maintain. So a lot of geneticists use them because you have a short reproduction cycle. You can look at various generations and genes passing through those generations very quickly. And also they look, developmental biologists like them because they're clear for the first sort of stage of development. So you can image them. Also, they don't take a long time to mature, and so you can look at the development in sort of a short amount of time. Now, the other really cool thing about zebrafish is that they have hearts which develop essentially the same as a human heart in the early stages. So we can look at human cardiac development with the zebrafish. It's transparent, so we can image the heart developing through time. And because of the easy genetic manipulation, because they've got very short reproductive cycles, we can investigate congenital heart diseases and how the genes affect the development of the heart. And, you know, we can make some conclusions about human development because the heart essentially develops the same way as a human. And this is just a bit of a schematic about cardiac development. So essentially your heart, your heart starts as just a tube. This is where the atrium will form. This is a ventricle. We have what's called the bulbous cordus and the sinus venous, which is where the veins come in and the arteries come in here. And at about 23, 24 days, this structure folds. And this happens in zebrafish and happens in humans. And we get this structure here where we have the ventricle formed, the atrium formed, and the bulbous is here. And we have this kind of folded structure. The blood will come sort of this way and through there, and so it's like a folded tube. And this is the same in humans and same in fish. So this is sort of the stages of development they're looking at. And at around this time, this is where the valves start forming as well. So if you're going to get problems with your valves, you can look at them here. And one of the things that we look at is how the valves are forming and how the fluid mechanics within here affects how the valves form. Because there's mechanical transduction in the way the cells form and align and develop, which is caused by the shear in the blood flow. And so we want to maybe look at that. So I've got an example here of a really nice dynamic microscopy image of a zebrafish heart. So you can see this folded structure. The blood's coming in here, and then it's sort of going away from the page. This is actually a 3D image formed by scanning. So microscopes get a plane image, and we can scan through very quickly and generate a 3D image. The blood cells are tagged with a different fluorescence dye than the myocardium, which is the heart wall. So these are the individual heart wall cells in green, and we have the blood coming through. So you can really see the dynamics of how the heart's beating. and how that's working. So you can contrast this to the next image that I'm going to show you, or the next movie, which is a bright field image. So this is a bright field microscope movie of the same thing. So we can see the blood, we can see the heart wall, but you can see the power of the fluorescence in differentiating those different types of tissue. Also, in the bright field image, we have all these out of focus structures. This is imaged through a live fish, so you've got sort of a lot of, they're not completely clear. So you've got all these out of focus structures obscuring our image. So what can we do with images like this? Well, this is what I was talking about before, where we might want to combine a bright field and a fluorescence image so that we can see where our fluorescence is coming from within the greater anatomy. So you can see that? Yeah, so this is the heart wall and the heart and the heart wall there fluorescing, and that's overlaid with the bright field image. And what we can do with images like this is do some further post-processing, which gives us some quantitative data about what's happening in there. So actually we can do what's called PIV, and I used to teach it. I'm not doing it now because we're doing less lectures, but essentially we can track the motion of the blood cells, and you can measure the flow. So those arrows show the speed and direction of the flow within the heart over the cycle. So that's the blood coming in and then passing out through that. And then if you look closely, you can see there's some what's called retrograde flow. So in this phase, as the valve closes, there's a little bit of flow that comes back. That's pretty hard to see without this post-processing. And this is indicative of a problem in the development of the heart in that valve. And this is present in some humans as well. All right. Any questions? Okay. So today, hopefully you've learnt these things. Resolution, what is it? How it relates to magnification and numerical aperture. How we measure resolution. Modulation transfer functions. How to interpret it. And also some principles about bright field, dark field and fluorescence microscopes. So next week, we're going to get onto some more medical stuff rather than biological stuff. So we'll talk about X-ray imaging, how X-rays are produced, how they're detected and how they're used in medicine. And also I'm going to do a little bit about ultrasound imaging as well. So thanks very much. Any questions, come and ask. Have a good rest of your day. So in confocal microscopy, you basically scan the sample. So the sample sits on a little stage with a very high precision robot, I guess, and you can scan the sample. So you've got a depth of field which creates your imaging plane in a normal microscope anyway. In the confocal microscope, the illumination is actually a focus point of light. So you scan that through, and then you can scan the sample. So you're sort of passing your sample through the in-focus plane. And so this happens really quickly with this. It's going at 200-300 frames per second. So you're scanning the whole thing in 1/300th of a second, and also raster scanning. Yeah. So this is a really good microscope. Take it here. So this is like one of the fast microscopes.
Also, you talk about X-ray imaging, right? So X-ray imaging, how do you actually generate? How do you combine fluorescence?
So X-ray imaging. So X-ray imaging is.
I go down to the.
Are you talking about bright field imaging?
Standard 2. You said that you use like.
Yeah, I can go to the 3 as well, but below that, it's just way too.
So what is this anyways, John? I came in like a bit too
This is how you measure the amount of magnification.
Bright field, to X-rays.
Yeah, but then.
like a mini pavlova.
When you take a semi-med or something.
That's how you measure the magnification level.
Because I've seen a semi-med, like, you don't actually have, you have only gray.
So you use monochrome cameras because essentially how a color camera works, or usually a color camera works by you just split your pixel up into a red pixel, a green pixel, and a blue pixel, and so you're going to lose resolution. Your native pixel is essentially going to be bigger, sampling pitch is going to be larger, and so you don't want to do that. You want to have pixels. So use a monochrome camera, and then so this one here is actually is just false coloured. So there's two channels, and it's taken. This is sort of phase average. So there's two channels, and you take one grey set of images and then another grey set of images, and the fluorescence that you're generating is for the different structures within those two movies. You can just colour one green, colour one red, and then put them on.
Combine them together.
So most of those images are false colour because you have a you want to use a monochrome camera because you don't want to be taking 3 sets of pixels at once. Yeah. So most scientific cameras are monochrome cameras, and you just create a false coloured image.
Yeah, but then like when you, for example, you're writing a paper on something you're observing.
Yeah.
So if you actually kind of manipulate, isn't that some sort of
Yeah. Yeah. So often these are often done kind of. A lot of papers are done, by people who just use the cameras only.
Yeah.
So it's sort of built within the software.
Yeah.
And the cameraman, the microscope manufacturer sort of does it for you. Yeah. But these is usually you do it as a representative colour of what the fluorescence is. So this does glow green.
Yeah.
Well, you don't image it in green. It is actually that colour. Usually. Yeah. Yeah. Yeah, and same with this. So this nucleus, it's called blue dappy, and you colour it blue. So if you're looking down a kind of an eyepiece, you'd see it blue. So it is kind of a bit, but yeah, that's just how they do it. yeah.
Also, I had a doubt of the project as well. So with the project, so the thing is with the requirements, as you mentioned for the requirements. So you do you have to come up with a table? Like, do you have to make a table? I made a table like this. So is that sufficient? Like, do you have to make a table just with these factors because from the
pretty much. And so it's hard to get it all in the like 20 on one page, especially with a bit of a description. So essentially, what you want to do is have a pretty concise description of why you set that requirement as it is. It could be just a couple of words. It doesn't have to be a huge
Also, I was looking into the requirements for my inhaler device. So I actually found one. It was a regulatory requirement that I was looking for. But the thing is, I wasn't getting access to that regulatory file. So do you know any database that I can search where I can get that?
Get the actual standard or something.
Yeah.
Monash has a lot of standards. For a regulatory requirement, I would you can just put the like one thing you could do is just say it must conform to ISO 13485 standard or something. And then that comes with a lot of requirements, but just to show that you've kind of looked into what the legal requirements might be. You don't have to.
So
you've only got 20.
I don't want to describe them as in like what them or mentioned in the standard. Like if I can just name the standard.
Yeah. Particularly if that's a required standard. Because you've only got 22, so you just need to pull out the main ones. And it's more about just writing a good requirement and making sure you're covering a range of kind of the important functional parameters.
Also, what's the importance of that? This code that you mentioned, what is the actual importance of it in a real case?
ID number?
Yeah, the traceability, the ID number.
Right, so if you remember the Woolies lecture where you showed you the V model.
Yeah.
So basically, you want to be able to identify each requirement at all stages, and what you have is you have some user requirements, and they will map to some functional requirements.
Yeah.
And so the reason to do that is that if one of your requirements changes, you want to be able to map that to all your other requirements. So if you have a certain user requirement, say that something, maybe some sort of legal requirement, that might change. So that might change the law. Now you've got this requirements changed. Now you need to be able to know what functional requirements that affects, because you need to then go and redo those, and then you need to, at every stage in the process, you need to be able to trace that requirement through. And so if you just have it numbered as if you don't have a number on it, it's just a list of requirements, how do you know
which one?
How do you need to map them together? Yeah.
So when you're actually designing as a product, so you got to link back every one of those.
So maybe say you've done everything, you've tested everything, you've got a battery,
Yeah.
which is 800 milliamps,
Yeah.
milliamp hours. Now, your supplier goes bankrupt, you can no longer get that battery.
Okay.
Now, you need to say, okay, now that's invalid. I haven't met that requirement anymore.
Yeah.
That's a functional requirement. But that's come about because I've got some user requirement. Some user said, I need a certain battery life. And so you say, okay, now that's not, so then you need to go and test your other requirements. You don't want to test all of them, you just want to test the ones that's affected.
Yeah.
So you go, okay, I've got this other battery. Now I need to test if I'm still meeting all my requirements. So I've changed that spec. What requirements does that relate to? And have I still met those requirements? Do I need to change those, change the spec, retest? Yeah. So it all has to link together, and that's about quality, quality control and quality assurance in design. And the standards are all particularly important for medical device because the stakes are higher, right?
Yeah.
And so you really need to conform to those standards. Because if you change the battery and all of a sudden your pacemaker doesn't work for the right time, then you kill someone. So it's a big the standards for that kind of thing for medical devices is much higher than so you've got ISO 9001, probably heard of, and that's just for any device, and then you've got 13485, which is the medical device standards, and the amount of testing and tracing and that kind of thing for that is much higher for ISO 9001. They're very similar, but there's just extra stuff you've got to do for medical.
Yeah.
Yeah.
Thank you.
Cool, no worries.